Hello students, a warm welcome to all of you in ITLS Academy. In today's session, we will discuss about the pharmaceutical biotechnology. In today's session, we will discuss what is the role of bioinformatics in a field of pharmaceutical and how combinedly this field will work. Okay, so we start today's lecture and understand. First, we understand what is a pharmaceutical biotechnology. Pharmaceutical biotechnology is a branch of science that apply the principle of biotechnology to develop a drug, vaccine, diagnostic and therapeutic agent. For what? To treat or to prevent the human diseases. Okay. What is a pharmaceutical biotechnology? Pharmaceutical biotechnology is a branch where we apply the principle of biotechnology. Means we apply the principle of molecular biology, we apply the principle of microbiology, biochemistry, genetic engineering, pharmaceutical science, and one and most important field, that is the bioinformatics. Okay, we apply the concept of bioinformatics, or we use a number of bioinformatics tool during the drug designing, vaccine designing, therapeutic and discovery, and develop a therapeutic agent like vaccine. This vaccine is a web tool basically that are used to find out the antigenicity of your molecule. Blast. Blast is a software that are used to find out the similar region within your what is a blast? Next is a blast. What is a blast? Blast is a basic local alignment search tool that are used to find out the similar regions. Okay. Next is a Clustal Omega. What is this Clustal Omega? Clustal Omega is a tool that are used for multiple sequence alignment. Emboss. Emboss is a software that are used for the pairwise sequence alignment. Swiss target prediction. Swiss target prediction is a software that are used to find out your target molecule. Swiss target prediction. Swiss admin. Swiss admin is a software that are used to find out the pharmacological property of your protein, of your target therapeutic agent. ToxicPred, a software available in a bioinformatics that are used to find out the toxicity of your proteins. Okay, and why we apply the concept of biotechnology for the drug development, for the vaccine development? Because we want to increase the production with high efficiency and efficacy. Okay, this is the importance of biotechnology in a field of pharmaceutical. What is the importance? Importance is to overcome the problem of traditional method, or to fulfill the demand of modern. Okay, the main purpose of pharmaceutical biotechnology is to apply the concept of biotechnology to develop a drug, vaccine, diagnostic and therapeutic agent. Okay, now we understand the history of pharmaceutical biotechnology. The history started before the 80s. Okay, and this era is known as the ancient and traditional era. In this traditional era, we use our natural resources for the therapeutic agent, or for the vaccine, or for the drug test, for the medicine. Next, we discuss the history of pharmaceutical. Before the 80s. Okay, ancient, and this era is known as the ancient and traditional. Okay, in this era we use our natural sources for the medicines. We use herbal extract, animal products and minerals as a therapeutic, and these are. Next is the pharmaceutical in food and medicines. Ancient civilizations used the fermentation process for the production of beer, wine, vinegar, and yogurt. Okay, later this become a foundation of industrial microbiology. Okay, in present it plays a most important role in a food industry. Industrial microbiology is also a branch and it plays a most important role in a food industry. Okay, next is the medical and practices were empirical without knowledge of microbes or genetics. Okay, next is the classical era or era between the 18th to 19th. Okay, in this era number of discoverable like Louis Pasteur. Louis Pasteur is known as a father of microbiology. George Mendel is known as a father of genetics. Louis Pasteur give the Louis Pasteur domesticate the fermentation process. Fermentation process is performed by the microorganisms. Okay, in 1865, George Mendel gave the law of heredity. There he proposed the three law of heredity. First is the law of dominance, second is the law of segregation, and third is the law of independent assortment. Okay. Next in 1882, Robert Koch developed a microbacterium tuberculosis, and this microbacterium tuberculosis is a microorganism that are responsible to cause a disease that is known as a TB. Okay. Next is the 1897, Edward Bunch show cell-free extraction method. Okay. Next, in a field of medical, vaccine was developed. Like Louis Pasteur developed a rabies vaccine. Jenner discovered the smallpox vaccine. Okay. Use of microbiology for producing organic compounds, alcohol and enzymes. Okay. Now, after the 19th, antibiotic and industrial era, 1920s to 1960s. Okay. In 1928, Alexander Fleming discovered the antibiotic, and the name of this antibiotic is a penicillin, and this is the first true antibiotic. In 1930s to 40s, industrial scale production of penicillin during the World War II. In 1944, Avery MacLeod and McCarty demonstrated that DNA is a genetic material. Genetic material means DNA is transferred one generation to another generation. Okay. During this experiment, these scientists use the radioactive compounds. Okay. Next is 1953. 1953, Watson Crick discovered the double helical structure of DNA. Okay. For giving this structure, they use the Next, in 1953, Watson and Crick discovered the double helical structure of DNA. DNA have a double helical structure like this. Okay. Next is the pharmaceutical impact, birth of antibiotic industry. Antibiotic industry means penicillin is discovered, streptomycin is discovered, tetramycin is discovered, and these all are the antibiotics. Okay. Fermentation industry expand to vitamins, to amino acid, or to steroid. Now present era. In present era, number of discovery will happen. rDNA technology is discovered, monoclonal antibody, genomics and molecular biology, pharmaceutical product and products introduced. Okay, in rDNA technology, we enhance the production of our gene. We enhance the quality of our gene by the help of cloning process. Okay, monoclonal antibodies, but we apply the concept of hydroma technology to develop a monoclonal antibody. Genomics means PCR is developed. Genomic mapping is possible. Sequencing is possible. Sequencing, number of sequencing methods are available, like Sanger sequencing, Max Gilbert sequencing, nanopore sequencing, Illumina sequencing, 454 sequencing, 6S RNA sequencing. Number of sequencing is developed. Okay. Recombinant insulin is developed. Growth hormones was developed. Clotting factor, interferons, cytokines, number of product is introduced. Okay. Next, modern biotechnology. Modern biotechnology first is a and in modern biotechnology, a most important technology is also developed, that is a CRISPR. And this CRISPR is also a gene editing technology, and it plays a most important role. Now we discuss the scope of pharmaceutical biotechnology. First is in a field of drug discovery and drug development. Biotechnology plays a major role to identify the new drug target, produce a biopharmaceutical drug by using a bacteria, yeast, and mammalian cells. By the help of rDNA technology, we enhance the production of therapeutic proteins. Here we apply the concept of fermentation and bioprocess and biochemical engineering. Okay. Next is your enzymobiology. Enzymobiology, we synthesize the enzyme. Okay. And these are the chiral intermediate in a drug. And the example is recombinant human insulin, monoclonal antibody, interferons. Okay. And here, for the drug discovery and drug development, we use a bioinformatics tools. Like PubChem, PDB, SIS, drug designing software, ToxicPred, PKCSM, ChemSar, ChemDes, etc. Okay. Next in vaccine designing. Vaccine designing. Pharmaceutical biotechnology is revolutionized the field of immunization. Why? Developing a safe and more effective vaccine. Like there are different types of vaccine, like live attenuated vaccine, conjugated vaccine, DNA and mRNA based vaccine, subunit based vaccine, polysaccharide based vaccine. Number of vaccine are available in today's. In today's scenario, number of vaccines are available, like recombinant vaccine, and the example is a hepatitis B vaccine. DNA and RNA based vaccine, COVID-19 vaccine. Edible and subunit vaccine under the research of polio and cholera. Okay. And the advantages of this vaccine: fewer side effects, higher specificity, and quick production during the pandemics. And also enhance the secondary response. And this vaccine acts as a immune booster. Okay. In bioinformatics, we use a tools like we use a database like NCBI, ENSMBL, and Baxygen, Alatop, etc. Okay, these are the bioinformatics tools that are used in vaccine design. Gene therapy and genetic engineering means We treat the genetic disorder, and the most important technology is CRISPR. CRISPR is a precision and genome editing technology. Gene replacement technology, viral and non-viral vector gene replacement technology. Here we apply the concept of homologous recombination. Okay. In CRISPR-Cas technology, this is also a gene editing technology. Here we apply three concepts for the replacement or for the knockout. For the knockout, we use the concept of non-homologous recombination. Okay. For knock-in, we use a concept of homologous recombination. Okay. And the basic principle of CRISPR-Cas technology is based on a basic principle of bacterial adaptive immune system. Okay. Viral and non-viral vector. Here we use a viral like bacteriophage as a vector to deliver our gene of interest. And the example is cystic fibrosis, hemophilia, cancer, muscular dystrophy, etc. Now, production of pharmaceuticals like biopharmaceuticals and therapeutic products derived from the biological resources like recombinant protein, insulin and interferons, monoclonal antibody, hormones and enzymes. Okay. And what is the advantage over the traditional method? These medicines are more effective, have a low side effect, and potential to cure and previously untreatable diseases. Okay. Diagnostic field like PCR probe, DNA probes, PCR, PCR basically RT-PCR or quantitative PCR. Electron DNA probe, here we use a number of probes like we tag the oligonucleotide with fluorophore molecule, with chemiluminescence molecules or with radioactive molecules to diagnose your diseases. Biosensors and immunoassay detect the biomarker for diseases like cancer and diabetes. Microarray and biochip technology this plays a most important role in personalized diagnostic. Microchips plays a most important role to find out the gene expression. Okay. Now we understand the key technology used in pharmaceutical biotechnology. First is the recombinant DNA technology. What is the recombinant DNA technology? In recombinant DNA technology, there are two main components. First is your vector, and second is your gene of interest. This is the vector and this is your gene of interest. You simply treat with this gene of interest and vector with restriction enzyme. What is this restriction enzyme? Restriction enzyme is a type of enzyme. It will cut the DNA at a specific position. Okay. Then we perform the ligation process. In ligation process, our gene of enzyme is ligated into the vector, and this process is performed by the enzyme that is the DNA ligase. Then finally transfer into the host cell. For this process, we use a concept of transformation. Okay, transformation, and this will transfer into the host cell, and here it will divide or multiply, increase the production of your gene. First is the isolation from donor organisms, then insert into the vector like plasmid, introduce into the host cell. Is host cell may be your bacteria, maybe your yeast, maybe your mammalian cells. Express the recombinant protein. And finally, purification and formulation of the medical uses. This is the recombinant DNA technology. Okay. By the help of this technology, we produce human insulin, growth hormones, clotting factors, vaccine, and enzymes. And this is the most and very important. Okay. Next is the hydroma technology. What is the hydroma technology? In hydroma technology, we produce a monoclonal antibody. What is the monoclonal antibody? This monoclonal antibody only are designed to treat a specific antigen. Specific antigen. What is the hydroma technology? Hydroma technology is used to produce a monoclonal antibody. What is the monoclonal antibody? Monoclonal antibody is an identical antibody that are used to treat a specific antigen. Like we isolate the B lymphocyte cells and myeloma cells. Myeloma cells is from the cancers and B lymphocyte cell for the antibiotic production. Okay. First, we isolate the B lymphocytes and myeloma cells. Then we fuse this B lymphocytes as well as myeloma. B lymphocytes and myeloma. Now fuse this. Now this is the hybrid cells. Okay. And this is known as a hydromas. Okay. And hydromas continuously produce a large quantity of single antibiotics. Okay. And the application is for the cancer therapy, for diagnostic to treat the autoimmune diseases. Okay. This is the hydroma technology. Next is your PCR. In PCR, this is a cell-free method for the amplification. This is the here we take the DNA. First step is the denaturation. This double stranded DNA is converted into the single stranded. Then aniline. In aniline, primers are bound. Then extension is new DNA is synthesized. Okay. For the PCR, number of PCR are available like normal PCR, quantitative PCR, RT-PCR, anchor protein PCR, hot star PCR. Number of PCR technologies are available. Okay, and this PCR plays a most important role in gene cloning, in sequencing, forensic and paternity tests, and diagnostic to diseases and infection. Now in protein engineering. Protein engineering means protein engineering involves the modification of structure to improve the stability and specificity of therapeutic activity. Okay. For this, we use a software like Swiss Biosphere, okay, to manipulate the structure of your protein. Okay, and technique that is the site direct mutagenesis, change the specific amino acids in your proteins. Directed evolution means here we mimic the natural select to optimize your protein. And the application of this protein engineering, long-acting immune analogs, insulin analogs, enzyme therapy, and biobetters. Designing the biobetter. What is the biobetter? Biobetter means improved version of existing drug. Now fermentation. In fermentation, we perform the culturing process. Means we culture the microorganisms to produce your biopharmaceutical. Okay. biopharmaceutical. Next is a fermentation technology. Fermentation technology means it is a large scale culturing of microorganisms or cells. For what? To produce a biopharmaceuticals under the controlled environment or controlled conditions. Here we use a bioreactor. In bioreactor, we grow our microorganisms and these microorganisms produce a primary metabolites or secondary metabolites. Okay, and the secondary metabolites are produced in a stationary phase and primary metabolites are produced in a log phase. Log phase. Okay, and there are different types of fermentation like batch fermentation, like continuous fermentation, like fed batch fermentation process. Okay, in batch, first we give the input and after the process we find the output. In continuous, we give the input and find the output continuously. In fed batch, we give the time to time or between a time interval and find after the whenever the process is completely complete, then we find the result. Means input and final reaction is when the process is completed, then we find the result. In continuous, input and output is given continuously. In fed batch, we introduce into a time interval or whenever this process will completed, then we find the result. Okay. And the application is to production of antibiotics, vaccine, enzyme, recombinant proteins. Next is your genomics and proteomics. Genomics is the branch where we study about the entire genome. What is the entire genome? Entire genome is a complete genetic material of an organism. Proteomics, in proteomics we study about the proteins. Okay, study all the protein expressed in cell, in organisms to understand the pathway and therapeutic response. And the application is personalized medicines and precision medicine, identification of biomarker for diagnosis and prognosis and target therapy. Okay. Now cell culture technology. In cell culture technology, we grow the animal and microbial cells in artificial environment. For what? To production of vaccine, to production of antibiotics, and to production of recombinant proteins. Okay, and the type is bacterial culture whenever we target the bacteria. Yeast and mammalian cell yeast and mammalian cell culture for complex glycoprotein and antibiotic. Application for vaccine manufacturing for large scale production of monoclonal antibodies. Okay. Downstream processing. Downstream processes means after the fermentation process, whenever the protein we found our target molecule or target therapeutic compound, then we purify that particular therapeutic compound, and the purification method is known as a downstream processing. Okay, in purification and recovery, after the purification, after the downstream process, we find the final result. Okay, and the step is cell separation. Let's suppose this is our fermentation and this is the fermentation broth where we find in this broth our material or our desired product will present. Okay, in this broth, first we perform the cell separation. We use centrifugation and filtration. Number of filtration techniques are available. After this filtration, then chromatography technique. Chromatography technique is also a technique that are used for the separation person. And there are a number of chromatography techniques are available like HPLC, GCMS, LCMS, ion exchange, size exclusive, and affinity chromatography. And finally, dry and formulations. Okay, now we find our desired therapeutic compound. Okay, and the application, this pharmaceutical gradient may be pure, safety and stable and safe for the biologics. Okay. Now for bioinformatics and computational biology. In bioinformatics and computer biology, in this branch we use a computer based tools. to analyze or to interpret the biological data. Okay, by the help of bioinformatics tool, we find out the gene sequences, protein structures, and drug interactions. Okay, like drug target identification, we use a software Swiss target prediction. For structure-based drug designing, we use a software web tool like CBDoc, Clustro, HeadDoc, PassDoc, etc. For molecular docking and simulation, we use Autodoc Bina, iMod, GrowMax, Ember, etc. AI-based drug delivery, where we use a concept of Swiss drug discovery softwares and clock discovery softwares. And AI drug based designing, here we apply the concept of artificial intelligence to develop a drug. Okay. Now, nanobiotechnology. We also integrate the nanoparticles with biotechnology to for the drug delivery and diagnostic purpose. For the drug delivery system, we use a dendritic cells nanoparticles for the drug delivery. Okay, and this plays a nanobiotechnology plays a most important role in target drug delivery to smart biosensors or nanocarriers. Okay. Now, advantages of pharmaceutical over the conventional medicine, like high specificity to reduce the side effect, can target the disease at a molecular and genetic levels, potential to treat the previously untreated conditions or personalized medicine. These are the advantages. Now, challenges: high cost, complex regulatory approvals, ensure the stability and storage and delivery, ethical concern, and development of resistance. Future: expense of biosimilars, growth of personalized and precise medicine, widespread of AI and machine learning, advancement in nanobiotechnology, and broad application in CRISPR. And this is all about today's session. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share, and subscribe ITLS Academy. Thank you.